turn the floor over to Norb for the con side of it.
Okay, I will be arguing the con. So, Reza, I didn't know you were going to use some of my publications against me. So I obviously will be arguing the con, and really the big question is, do animals provide a sufficient basis for presumption of toxicity and safety testing? And obviously I'm going to be arguing for the application of human cell-based assays in toxicity testing. And so some of the examples I'm going to be using are based on the immune system, but I think the comments I'm going to make, I think, are more broadly applicable. And so to first begin, you know, we've talked a lot about the fact that the target species that we're trying to do regulatory assessments for really is the human, and yet the fact is that we have very little human data and often have no human data at all. Even in the case where we have epidemiology studies, we've heard a lot about a lot of the issues around that. Perhaps one of the biggest is we don't even often know what the exposure level is. Not to mention all of the other things that were in the box that I think Lisa or somebody earlier showed with the big question mark. You know, the one area where we do have information comparing animals and human data are clinical trials for pharmaceuticals. And there we find that quite often the animal really does not replicate what the human response is. So historically in toxicology, due to ethical reasons, you know, we've chosen to do our studies in animals, and quite honestly, most of that data is in rodent models. And so the question really is the rodent, and that's what I'm going to argue today, is the rodent really a surrogate for the human? And what I've done is I've actually identified two studies that I want to discuss, which have been published somewhat recently. And the first one I want to talk about was actually a very large study. You can see all the different authors on this paper. But what the point of the study was to compare genomic responses between human and the mouse in terms of various inflammatory stimuli. And what they did here was they compared three inflammatory stimuli: severe blunt trauma, burn injury, and low-dose bacterial endotoxin. Now, as you can imagine, if you do this kind of study, there's a lot of complexity around doing these human measurements. Obviously, the demographics, severity of injury, the clinical outcomes, and time to recovery. And so these studies were actually quite complex. So for example, for severe blunt trauma, 167 individuals or patients were followed with serial blood draws, and actually leukocytes were assessed here up to 28 days, and then, you know, the transcriptomic signature was determined in these studies. So for example, for severe blunt trauma, there were 4,389 gene changes compared to controls that were greater than twofold. In terms of burn injury, 244 individuals were studied up to 1 year, again doing serial follow-up measurements. In this case, 2,251 gene changes were observed. And also kind of the classic inflammatory stimulus, bacterial endotoxin was actually studied in 4 individuals. Short-term responses, 24 hours, and again, there were 2,251 gene changes. And so the comparisons that were made in this study were human to human in terms of different stimuli, mouse to mouse in terms of different stimuli, in terms of the genes that were expressed, as well as across the species. And so what did they find? Well, they found that when comparing the correlation of gene expression changes in humans, especially if you take a look in the first box here, looking at human the burn injury compared to trauma in humans, very good correlation in terms of the gene expression changes. In spite of all of the caveats I just mentioned about demographics, severity of injury, and so forth. If we compare endotoxin to, let's see, what do we have here? I can't see from here. Trauma, you can see there's still moderate correlation. Same for burn trauma and endotoxemia here. And so there's moderate correlation. But what's really striking is when you start comparing the mouse. So if you compare mouse endotoxemia to mouse trauma, there's no correlation. If we compare mouse endotoxemia, for example, to the gene expression pattern resulting from burn injury as a stress stimulus, also no correlation. And more telling is when you start comparing across species. So let's compare, for example, human burn to mouse burn, which is here, virtually no correlation. And if you look at this part of the table, these are really the cross-species correlations. So there's really very, very little, if any, correlation across these two species. And in fact, the authors concluded that among genes changed significantly in humans, the murine orthologs are close to random in matching their human counterparts. So let me show you another study. This is actually one in my laboratory where we compared human primary B cells from 3 species. So we did human, mouse, and rat. And we wanted to make the comparison as close as possible. So not only did we purify B cells, we went the extra step. We took naive B cells from all 3 species because, as you know, rodents, when we house them at our universities and our laboratories, they're pretty much isolated from any kind of pathogens. So virtually most of the immune cells in these animals are naive. So we went the extra step and purified these naive cells. And then we activated them in the identical manner, and that was in the presence and absence of TCDD. And during a 24-hour time period, we isolated cells at 4, 8, and 24 hours, and then we looked at the gene expression profile. And you can see in human primary B cells, we had differential expression of about 554 genes. In the mouse, we had a whopping over 2,500. In the rat, we were around 700, almost 800. What we were interested is how did the 3 species compare in terms of their gene expression profile? And so the answer really is here within the 3 circles. We had 28 genes that actually were differentially expressed across these 3 species. And in fact, of those 28, I would argue that probably very few of them even had anything to do with B cell function because they were the AH receptor battery genes like cytochrome P450 1A1, 1B1, some of the transferases and so forth. So again, it shows you how little there is in terms of similarity in terms of these gene profiles. So you might ask yourself the question, why is that the case? Well, if we start looking at what has been the ENCODE project, and so for those of you, I'm sure most of you are familiar with this, but for those that are not, basically the goal of ENCODE was to map functional elements of the human genome. So in large part is looking at the regulatory regions of these genes. And as this was going forward, a second initiative called the Mouse ENCODE was also started. And there was a series of 3 papers that were published in Nature 2014 that began looking at comparisons across the 2 species. And so I just selected this one cartoon which summarizes, I think, some of the really important findings from this paper, or these 3 papers. So the first thing is when you begin to compare across species and look at orthologous genes, the first thing you see is that the level of transcriptional activity is actually quite different for many of these genes. If you begin to look at the regulatory regions, you find, and again, this is kind of illustrated in this cartoon, that some of the transcription factor binding elements or sites are positioned in different places when you look across these species. And in fact, many of the transcription factors that drive these orthologous genes have binding sites in one species, but not in the other. Also, if we look at the enhancer region, we see the same thing. Different transcription factors are regulating these regulatory regions of the genome. And in fact, to add even further complexity, we have these retrotransposons, which basically are DNA that has been converted to RNA and then reverse transcribed and spliced back into the genome. Well, these also have transcription factor binding sites, which further change or alter the regulatory regions of these genes. So we should not be that surprised that there are differences between, for example, the mouse and the human. These two species diverged somewhere between 70 to 75 million years ago. So let me finish with this slide. I think you've probably seen this quote a number of times. It's by George Box that basically he says essentially all models are wrong, but some are useful. Animal models, especially rodents, I think may be poor surrogates of human biological responses to exogenous stimuli, and these two papers I cite are examples of that. And so, you know, what's the path forward? And I guess this is part of the discussion later on, but I'll just throw this out now. You know, employment of human cells and tissues when possible as an adjunct to animal testing, I think is very, very important. Development of human synthetic in vitro models. I think Reza and I will have an opportunity to debate some of the things he shared in his presentation. So, but the fact is that we do need better in vitro models, and certainly the in vitro models we have now do not simulate all the aspects of what happens in the intact organism. And then finally, I think some of the new genomic information we're obtaining helps explain a lot of the difference that we're seeing across species and gives us a lot of information about mechanism. So with that, I'll stop and turn this back over, I guess, to Gary.